It's true. Cells may be considered to be the unit of life. This means that many of the processes essential for life are carried out inside cells. And cytology is the term we use to describe the study of cells. And a cell really is the smallest unit that can be considered to be alive. And some simple organisms, such as the amoeba, are composed of a single cell. But in humans, many cells work together. And a genuine scientific estimate of the number of cells in the human body has come up with 37.2 trillion cells. So in your body there's 37.2 trillion cells, all working together. Now these cells are different, they're differentiated, but they have many things in common. And that's what we want to think about in this talk. Now, the first person ever to see cells under a light microscope was a guy called Robert Hooke. He was an English scientist and he'd built himself a new light-based microscope. And he looked at some cork down the microscope. And, of course, this is a plant tissue, so he was looking at plant cells. And we now know that plant cells are surrounded by a cellulose cell wall. So looking at plant tissue. And he saw that the cells contained box-like compartments. And these reminded him of the cells in a monastery that monks sleep in, because that was the original meaning of the term cell, so where a monk sleeps. But he called them cells because that's what they reminded him of. So Robert Hooke coined the term cells in 1678. Now, animal cells, and of course humans are animals, don't have a cellulose cell wall. So in a light microscope, with relatively low magnifications of just maybe 100 or 2, you can only see fairly basic components. Round about the outside there's a cell membrane which is compartmentalising the cell from its environment. Then underneath that there's an area called the cytoplasm. And then within the centre of the cell there's an area called the nucleus. And these are very hard to see without staining. So very often these have to be stained even to see these components under a light microscope. But bear in mind that cells are only 5 to 10 micrometers. A micrometer, of course, is 1,000th of a millimetre in size. So they can only be seen with the aid of a microscope. They are microscopic. And this is what we see under a light microscope with relatively low levels of magnification. Now, with an electron microscope, things get a lot more interesting. Because when we use an electron microscope, we are using a form of electromagnetic radiation, electrons, which have a much smaller wavelength than light. So light has a large wave amplitude, whereas electrons have a small wave amplitude. So that means the smaller wave amplitude of the electrons can penetrate much smaller structures, giving us much greater detail than a light microscope. And when cells are examined under the higher power of an electron microscope, the organelles can be seen. And these organelles are the subunits or the components of the cell. And in the same way that the body is made up of organs, the functional units, a cell is composed of organelles, which are the functional units of the cell. So organelles are the structures within the cytoplasm, and they perform specific functions. They are the functional units of the cell. Let's start off by thinking about the cell membrane. This is a very thin membrane which surrounds the outside of the cell and marks the boundaries of the cell. Now, cell membranes are composed of lipids, phosphates and proteins. And because the cell membrane is composed of 2 layers of phospholipid molecules, it's referred to as a phospholipid bilayer. That's why you can see on this diagram it has 2 layers to the external cell membrane. And a phospholipid molecule is made up of a combination of phosphate and fatty acids. And cell membranes also contain some cholesterol. The external cell membrane compartmentalizes the cell and therefore controls movement of fluids and other substances in and out of the cell. And this regulatory function means the cell is able to control, to a fairly large extent, its own internal environment. Some of the proteins in the cell membrane are referred to as transmembrane proteins. This means they span the membrane from outside to inside. They have ends in the external tissue fluid and internal cytosol, the cytosol being the fluid in the cytoplasm. Some of these proteins regulate the passage of substances in and out of the cell. For example, some transmembrane proteins act as tunnels to allow glucose molecules from the tissue fluid into the cytosol, where, of course, once they're inside, they can be used by the mitochondria to produce energy. Other membrane-bound proteins act as receptors. And these specialised receptor protein molecules allow the cell to recognise chemical messengers from other cells or from endocrine glands located in completely different parts of the body. Once a receptor has detected the presence of a particular chemical messenger, this will initiate some physiological change within the cell. As a result, cells do not function in isolation, but as a coordinated and disciplined component of the whole body. Now, we mentioned the cytosol, cyto, cell, sol, solution. And this is the fluid found in the cytoplasm of the cell. It is the intracellular fluid, inside the cell. And the cytosol is mostly water with some additional dissolved and suspended molecules. And the combination of the molecules and the water, because it's large molecules that are suspended in the water very often, it's referred to as a colloid. So the cytosol is a colloidal fluid. And organelles are suspended in the cytosol. And much of the water in the body is located in the cytosol as well. So we see the cytosol has different functions here. It is padding out the cell. It is the medium in which the organelles are found. And it's also the major fluid reserve of the body. And many biochemical reactions occur in the cytosol, such as the initial processing of glucose in the production of energy and the synthesis of fatty acids. So it's providing a location for life-giving biochemical reactions. Now we notice that the cell has a cytoskeleton, cytocell. So cells have their own skeletal structure, and these are microfilaments and microtubules, and they're composed of proteins, and they extend throughout the cytosol. And collectively, these structures form an internal skeleton within the cell, the cytoskeleton. And the cytoskeleton provides structural support and strength for a cell, and helps to maintain its overall shape. And cell movement can also be facilitated by the cytoskeleton. And this is necessary, for example, during early embryonic development of the body, where cells must migrate into the correct positions to form the organs and the tissues of the body. And movement is also important for muscle contraction and the movement of white blood cells to combat infection. So several roles of the internal cytoskeleton. The endoplasmic reticulum is an extensive network of flattened, interconnected channels and tubules distributed throughout the cytoplasm. An endoplasmic reticulum is itself bounded by membranes. In fact, it is a series of these membranes. And the functions of this extensive structure is to move material around inside the cell. Intracellular translocation, translocating material, communicating material around the cell. As different organelles within a cell perform different functions, particular substances will be required in particular locations, just like in any other factory, because we can think of the cell as a biological factory, the unit of life. Material needs to be moved around to be in the right place and the right time. And in this sense, the endoplasmic reticulum is analogous to a road or a rail network, delivering raw materials and transporting away the finished products. And in addition, the endoplasmic reticulum supplies support for the overall structure and shape of the cell. Now, the ribosomes, which we'll come on to in a minute, are often associated with the endoplasmic reticulum. Now, if an area of endoplasmic reticulum is associated with ribosomes, it's described as being rough endoplasmic reticulum. Areas without associated ribosomes are termed smooth endoplasmic reticulum. So rough endoplasmic reticulum we can see on this diagram with ribosomes, smooth endoplasmic reticulum without ribosomes. So next we need to learn what these ribosomes are all about. So we see these small ribosomes associated with a rough endoplasmic reticulum. And these are small, dark staining organelles. Now, I've said they're often associated with rough endoplasmic reticulum, but there are free ribosomes sometimes found in the cell in other parts of the cytoplasm as well. And the rough endoplasmic reticulum is rough precisely because it is studded with these ribosomes. Now, the ribosomes receive genetic instructions from the DNA, that is the deoxyribonucleic acid, which is located in the nucleus of the cell. And this information, this genetic information, is then transferred to molecules of RNA, ribonucleic acid, which then string together long chains of amino acids to form polypeptides and proteins. Because amino acids are the subunits of proteins in the same way that bricks are the subunits of a wall. And ribosomes are therefore the site of protein synthesis. In essence, they are little protein factories. Now, some of the synthesized proteins are used to form the structures of the individual cell, whereas other synthesized proteins will be exported from the cell for extracellular purposes. So if you think about it, it's logical that the ribosomes are usually associated with the endoplasmic reticulum, so that they can be supplied with amino acids, which are the building blocks of the protein, and then synthesized proteins can then be removed to where they are required. So the ribosomes are synthesizing these big biological molecules. They are synthesizing these proteins. Now, the Golgi apparatus, or the Golgi complex, is an arrangement of membranes which are responsible for the export of products from a cell. So products from the endoplasmic reticulum are transported to the Golgi, where some further molecular modification of the produced substance often takes place. And once the fat or protein-based product is ready, so-called secretory vesicles bud off from the main Golgi complex and transport the material to the external cell membrane for export from the cell. So, for example, some cells in the digestive system secrete digestive enzymes in this way. Endocrine cells secrete endocrine hormones, and liver cells secrete plasma proteins. So the material produced inside the cell is excreted or secreted from the cell to be used in an environment external to that cell itself. Now, one of the absolutely fundamental properties of life is that life is able to synthesize its own energy requirements. And the energy inside cells is produced by the mitochondria. So there are several mitochondria. A single mitochondria would be referred to as a mitochondrion. And these are kind of sausage-shaped structures. And they have an outer membrane and a highly enfolded inner membrane. And this enfolded inner membrane provides a large surface area for the location of enzymes, which are responsible for the generation of energy within the cell. These enzymes produce energy by using oxygen. They use oxygen to oxidize or burn fuels such as glucose or fatty acids. And the energy actually comes from breaking the chemical bonds which hold together these fuel molecules. That is the glucose molecules or the fatty molecule or whatever it's metabolizing at the time. And all of the fuel required for energy production derives from food. And all of the oxygen, of course, is from breathing, from the atmosphere. Now, mitochondria are therefore the site of all energy production, and they're often referred to as the powerhouse of the cell. So it's amazing, all the energy being used in the cell comes from these mitochondria. And cells which use a lot of energy, such as those in the muscles, which of course are actively contracting, have a lot of mitochondria in their cytoplasm. Whereas cells which need less energy, such as those in the skin, for example, have fewer mitochondria. Liver cells and kidney cells are also very metabolically active, so they'll have large numbers of mitochondria in their cytoplasm. And the reason cyanide is such a fast-acting poison is because it halts the process of energy production in the mitochondria. And if the mitochondria no longer produce the energy, if that's been blocked by the cyanide, then no life-giving processes can be fueled. Therefore, there will be no physiology and there will be a sudden cessation of life. And it's interesting that mitochondria also contain some DNA, some deoxyribonucleic acid. And this mitochondrial DNA is only passed on through the maternal line. So from mothers to daughters and from mothers to sons. But of course, with the son, that mitochondrial DNA will die with the individual. It's only passed on by the daughters through the maternal line. Lysosomes are organelles surrounded by a typical membrane, and they contain a variety of digestive enzymes inside, such as lysozyme. So lysozyme is found in lysosomes. And they're formed by breaking away from the Golgi apparatus and then dispersing throughout the cytosol. And lysosomes are used in the process of intracellular digestion, that is, digestion within the cell. And the digestive enzymes in the lysosomes break up large complex molecules into smaller ones using the process of hydrolysis. And these hydrolyzing types of enzymes split large molecules up by adding hydrogen and oxygen. And this process of intracellular digestion may digest material from outside the cell. For example, in white blood cells, foreign material is ingested into the cell. Lysosomes approach the foreign material and spill their digestive enzymes onto it, therefore digesting it. So lysosomes are therefore essential in this process of phagocytosis. Phagocytosis being the process of cell eating. And in addition to this, digestion of extracellular material, as well as this, lysosomes may also digest intracellular material, that is, material derived from inside the cell itself. As cells need constant maintenance, new components and organelles are continuously being formed, meaning old ones need to be removed. So unwanted cellular organelles and biomolecules are digested by the lysosomal enzymes in the process of autophagy. Autophagy literally meaning self-eating. And lysosomes may also burst to digest the cell as a whole, if a whole cell is unwanted. And this process is sometimes called cell suicide. The process is correctly named apoptosis. Apoptosis is the digestion of a whole cell. Peroxisomes are organelles which are similar in appearance to lysosomes, but the peroxisomes are smaller. And peroxisomes contain enzymes, including hydrogen peroxide, which work by oxidizing substances. And if these toxins were not oxidized, they will build up to poisonous concentrations within the cell. For example, about half of the alcohol a person might drink is detoxified by peroxisomes in the liver. The nucleus is located at the centre of the cell, inside the cytoplasm. And there's a separate membrane surrounding the nucleus referred to as the nuclear membrane. So the nuclear membrane is compartmentalizing the nucleus and the cytoplasm. And within the nucleus there are the chromosomes. Chromo means coloured, somes means body. These coloured bodies, the chromosomes. And all human cells contain 46 of these chromosomes, 46 of these bodies, arranged into 23 pairs. And the chromosomes contain structural proteins called histone proteins, but they also contain the genes, which are the genetic information responsible for controlling the cell. The genes are composed of the DNA, the deoxyribonucleic acid, carrying the genetic code to make the cell, to make the tissues, to make the body. And this is why the nucleus is sometimes referred to as the control centre of the cell, because it's where the actual information is contained to make the cell. A nucleolus is a cluster of DNA, RNA and protein within the nucleus. And these are the same types of molecules found in the ribosomes. So the nucleolus is actually where the subunits of the ribosomes are synthesized before being exported to form the ribosomes in the cytoplasm. And once in the cytoplasm, the ribosomes, of course, will synthesize proteins. But the ribosomes themselves are actually synthesized in the nucleolus. During growth and development, cells differentiate into specific types, which allows them to perform particular functions. And this differentiation is initiated and regulated by genetic instructions from the genes, which carry all of the required information. Different cells must have a specialized structure, which results in the production of many complicated and different types of cells. And it's this process of specialization which is referred to as differentiation. So specialized cells are required to form the different types of tissues needed to construct the large structures and the organs of the body. Different types of cells include nerve cells or neurons, muscle cells, liver cells, blood cells, epithelial cells. Each cell has a structure and function specific to the role of a cell or tissue of which it is a member or a component. Groups of similar cells, of course, compose tissues, and groups of tissues compose the organs. So all of the cells might look quite different, but they do contain these same functional organelles. and yet they differentiate to perform the different functions required in the different tissues of the body. Now, of course, cells have to reproduce. Cells are derived from parent cells, which underwent a process of cell division. And in this process, one parent cell grows and then divides into two daughter cells. So this process is based on cell division. All cell reproduction is based on cell division. And when a cell divides, it produces two daughter cells. Even if you're male, they're still called daughter cells. And it's not too difficult because there only are two forms of cell division. There's mitosis and meiosis. Let's look at mitosis first. Now, mitosis is sometimes described as simple cell division, although it's not really a simple process. Now, during the non-dividing phase of the cell cycle, inside the cell it will duplicate a set of 46 chromosomes. So the 46 chromosomes will divide, each producing a new chromosome the same as itself. Each gene is precisely copied to make an exact copy of the original gene. And during mitosis, one complete set of chromosomes migrates to each end of the cell. So you start off with 46 chromosomes. Each chromosome makes a copy of itself. So we've now got two sets of 46 chromosomes. And one set of 46 chromosomes migrates to each pole of the cell. And this parent cell then divides into two daughter cells, which are going to each contain 46 chromosomes. And because the daughter cells and the parent cell all contain the same number of chromosomes, this form of cell division is sometimes referred to as conservation cell division. So the number of chromosomes is going to be conserved. The group of descendant cells from a parent cell is described as a cell line. And mitosis is so important. It's the type of cell division that occurs when the body grows from a zygote, which is the single fertilized cell, into a completely formed new baby. And it's also the form of cell division which generates new tissue in the process of wound healing. And in addition, ongoing mitosis maintains the health of most body tissues by replacing old cells with new ones. And because mitosis is the type of cell division that goes on in the body, it's also sometimes referred to as somatic cell division, because soma means body. So we can see that the parent cell here is producing the extra set of chromosomes. The nucleus is separating and dividing. So we have two separate nuclei, but each with its own set of 46 chromosomes. And then finally the cytoplasm pulls apart to produce two daughter cells, which can then grow on, and each of those can then divide via mitosis or mitotic cell division in turn. So mitosis is somatic cell division, somatic cell reproduction. The only other form is meiosis. So that's the only other type of cell division, and it's only used in the production of gametes, which are the male sperm and the female ovum. Now, the function of sperm and ova are to fuse together, forming fertilized cells. And such a fertilized cell is called a zygote. And once formed, a zygote undergoes repeated mitotic cell divisions using mitosis to form the new body of the fetus and the baby. Now, if you think about it, if the sperm and ova each contained 46 chromosomes, then the next zygote would contain 92, and the number of chromosomes would double every generation, which would clearly be absurd. So to prevent this happening, before reproduction takes place, there is this reduction cell division. And in the ova and in the testes, cells are produced using meiosis, which contain half the number of chromosomes. They contain one chromosome from each of the pairs of chromosomes. So in a diploid cell, in a somatic cell, there are 23 pairs of chromosomes, making 46. And in the gamete there's only one chromosome from each pair, making 23. Then the 23 from the sperm combined with the 23 from the ovum, restoring the diploid number of 46 again. 23 is referred to as the haploid number. So the sperm cells and the ova cells are haploid, all other body cells are diploid. This diagram puts these two forms of cell division together into the human life cycle. So we have an adult, and then there is meiosis to produce the sperm and the ova, each containing the 23 chromosomes. They combine together to form the zygote containing the 46 chromosomes, and then there's repeated mitosis, 2 cells, 4 cells, 8 cells, 16, 32, 64 cells. Every time there being a conservation cell division, the number of chromosomes being conserved at 46. That grows into baby, baby grows into the adult, all using mitosis. Along the way there's going to be injuries which are going to heal by mitotic cell division. Along the way there's going to be wear and tear, replaced by mitotic cell division. The cells which are damaged or worn out will be replaced by mitosis. Along the way, of course, there's going to be growth. That process is facilitated by mitosis, by an increase in the number of cells, until you get to the adult stage, and then meiosis will take place again, just to produce the gamete cells, the sperm and the ovum.